We continue our lecture, and next we are going to talk about microorganisms and infectious diseases. That means we're going to talk about what pathogens have to be able to do to cause infection. To cause infection, a pathogen must be able to, number one, maintain a reservoir. Reservoir means a place where pathogen goes before and after infection. Number two, pathogens have to be able to leave reservoir and enter the host body. That means here we're going to talk about entrance of infections and transmission of infections. Number three, pathogens have to be able to adhere, means to attach to the surface of the host cells. Number four, pathogens have to be able to invade body of the host, means some of them have to be able to go deeper in the tissues. Next one, pathogens must be able to evade body's defenses, means somehow go around our immune system. Number six, pathogens have to be able to multiply within body of the host. And number seven, they have to be able to leave body of the host and return to reservoir. But before we will go through all 7 virulent factors, let's talk about those two terms that we use a lot when we talk about infectious diseases. So what is it infection and what is it disease? Infection refers to growth of pathogens in the body. Disease, it is a consequences of the infection. But usually in everyday life we use those terms together. We say infectious disease. It means pathogens grow, multiply within the body of the patient and cause symptoms of infectious disease. First, a virulent factor. Pathogens have to be able to maintain a reservoir, means place where they go before and after they cause infection. The most common reservoirs for human pathogens are the other humans, animals, and the environment. So let's talk about first reservoir, human reservoirs. This group can be divided into two subgroups, sick individuals and healthy carriers. For example, a common cold is caused by viruses that are very fragile, and because they're so fragile, they're not able to survive outside of humans. So that's why sick individuals in this case will be the source of infection or reservoir. Healthy carriers, this type of reservoir can be also divided into two groups or subgroups, incubatory carriers and chronic carriers. Let's talk about incubatory carriers first. Each infection has what we call incubation period. It is a period of time between 2 points. The first point, this is when patient gets infected, and the second point when the same patient develops symptoms of infection. For different infectious diseases, incubation period can last for different period of time, from few hours up to few years, for example, as HIV infection. So during this incubation period, infected individual is a source of infection. And as I said, for example, for HIV infected person, incubation period can last for a few years. And then another example, those are chronic carriers. Chronic carriers. Some people, after they get infected, either develop symptoms and then recover but after that they become a chronic carriers of that pathogen. Or sometimes they get infected, and because they have strong enough immune system, they do not develop symptoms, but for some reason again they become carriers of that pathogen. Means they're going to be a source of infection. Second example of reservoir will be animal reservoirs. When animals are a source of infection and it can be transmitted to humans, those type of infections are called zoonoses. And of course the most common example of zoonosis will be rabies, when the source of infection will be wild or infected domestic animals. And the last example of reservoir will be environmental reservoirs. Those are soil and water. For example, soil is a source of Clostridium species. Please remember, Clostridium species are able to form endospores, and in this form they can be found in the soil. So we can say that soil is a source for all Clostridium species. And another environmental reservoir is water, and of course any gastrointestinal infection can be transmitted through contaminated water. Second virulent factor. Pathogens have to be able to leave a reservoir and enter the body of the host. It means next we are going to talk about portals of entry and modes of transmission. Let's start with the portals of entry. Portals of entry for infectious diseases will be body surfaces, mucous membranes of nose, mouth, conjunctiva, skin, mucous membranes of urethra, vagina, and placenta. Some pathogens use different portals of entry. For example, the usual portals of entry for sexually transmitted diseases are urethra of males and vagina of females. But it can be also a throat or rectum if these are points of sexual contact. The more pathogens enter the portal, the more likely infection becomes. So the question is, how do we measure infections? Let's look at those two terms that we have to know for your exam. The first one is ID 50 or infectious dose. It means number of the cells required to cause infection in 50% of individuals. And then LD50 or lethal dose. It is a number of cells required to cause death in 50% of individuals. Let's look at this picture on this chart on this slide. As you see, LD50 for Streptococcus pyogenes is about 50 cells, and LD50 for Salmonella typhi is about 8,000 cells. What does it mean? If there are only 50 cells required to cause death in 50% of individuals of Streptococcus pyogenes, it means Strep pyogenes is much more aggressive, much more virulent compared to Salmonella typhi. There are many ways that pathogens are carried to humans. This is what we call modes of transmission. The most common mode of transmission is respiratory droplets. For example, flu, meningitis, chickenpox, measles, all those infections are transmitted when infected patient cough, sneeze, laugh, or talk. The next mode of transmission is what we call fomites. Those are objects around us. Cups, towels, sponges, spoons, brushes. Usually, a lot of pathogens are not able to use fomites as a reservoir because they're not that stable, not that resistant. They're not able to survive on the surfaces for a long period of time. But with fomites, infections can be transmitted from one person to another person. Direct contact is the next mode of transmission. As an example, we can use sexually transmitted diseases. Herpetic infection caused by herpes simplex type 1 or type 2. And also vertical transmission also belongs to this group of transmission. And vertical transmission, it means from mother to child, and it can be prenatal or perinatal. Prenatal means pathogen is able to cross placenta. As an example, we can use Treponema pallidum. It causes syphilis. So if mother is infected with that bacterium, then Treponema pallidum crosses placenta, affects child, and if that pregnancy doesn't end up with abortion, then the child will be born with congenital syphilis. And perinatal, it means during the birth or shortly after the birth. Let's say mother has gonorrhea. When child goes through the birth canal, the mucous membranes of eyes of the child will be infected, and child will develop symptoms of gonorrheal conjunctivitis. The next mode of transmission is fecal-oral route, and this is the most common route of transmission for gastrointestinal infections. And it can be, again, direct or indirect. Direct means through the body contact. A very common route of transmission for medical workers. When a medical worker takes care about a patient with gastrointestinal infection and doesn't change gloves, wash hands, there is a chance for that person to get infected. Indirect means through contaminated food, water, fomites, or vectors. The next mode of transmission is arthropods or arthropod vectors. We already talked about arthropod vectors on one of our previous lectures, and remember, there are two types of vectors we have to know: mechanical vectors, for example, housefly, and the biological vectors, for example, Anopheles mosquito that transmits malaria. Next group of infection, we call them airborne infections because they are transmitted through the air. Of course, as an example, we can use Mycobacterium tuberculosis. It causes tuberculosis in humans, and it is an extremely resistant bacterium. It can stay in the air for few hours, and every time when healthy individuals inhale that bacterium, there is a chance that they get infected and might develop symptoms of infection. And the last one, the last mode of transmission is parenteral. It means direct entrance through the blood vessels, tissues below the skin, or mucous membranes. As an example, of course, we can use HIV infection. The next virulent factor is ability of the pathogens to adhere to the surfaces of the cells of the host. We already talked about adhesins on one of our previous lectures, and I told you those are proteins that are formed by certain bacteria, and using those adhesins or proteins, pathogens are able to recognize surface receptors on the cells, and then using those adherents, they attach to the surface of the cells. As an example, we can use Bordetella pertussis. You can see it on this picture on this slide. This bacterium causes pertussis in humans, and as you see, the target cell for Bordetella pertussis will be cilia of ciliated epithelium. So Bordetella pertussis has the ability to produce adhesins to receptors on the top of the cilia, and then it attaches to the surface of those cells, and it's very difficult for our immune system then to remove it from there. After attaching to a body surface, some pathogens stay where they attached. For example, Bordetella pertussis. It is attached to the cilia of ciliated epithelium and will stay there. So it means pertussis is not invasive infection. Pathogen doesn't go deeper in the tissues, doesn't penetrate deeper in the tissues. But there's another group of infections, we call them invasive infections. In this case, pathogens penetrate deeper into the tissues. As an example, we can use malaria. Remember, we talked about life cycle of malaria, and you remember that Plasmodium malaria actually uses liver cells as a first stop, and then it gets back into the bloodstream and then infects red blood cells. So that is an example of invasive infection. Another example of an invasive infection, of course, will be tuberculosis, because Mycobacterium tuberculosis can infect pretty much every system of human body. The next few pictures show actually how pathogens go deeper into the tissue and make that infection invasive. Let's look at the first picture. This is our pathogen, some kind of bacteria. Those are epithelial cells. That is what we call basement membrane, and those are deeper tissue over here, and blood vessels will be here on the bottom of this slide. So the first thing what pathogens do, they recognize surface receptors on the surface of the cell and then attach to those surface receptors. The second step, pathogen makes that cell to engulf itself. This way, pathogen gets into cytoplasm of that cell. As soon as it gets into cytoplasm of the cell, you see it covers itself with vacuole. Why does it cover itself? Because if it doesn't cover itself with the vacuole, it will not be protected against enzymes that can be found in cytoplasm of this cell. It means there's a possibility that it might be destroyed by those enzymes. So within that vacuole, pathogen starts moving towards the basement membrane. After pathogen reached actually bottom of the cell, it leaves that cell and then starts to reproduce and multiply beneath the epithelial cell. Then it destroys basement membrane and then goes deeper into the tissues, gets in the blood vessels, lymphatic circulation, means infection becomes systemic. The next virulent factor, pathogens have to be able to go around the immune system of individual. Let's start with the major host defense against pathogens, which is phagocytosis. Before phagocyte can phagocytize the microorganism, it must first come in direct contact with the surface of microbial cell. But some pathogens avoid such a contact by producing slippery mucoid capsule. Remember, I told you already, capsule is also called virulent factor of the pathogen. And I also told you that the main function of capsule is protection. And that explains why pathogens, very serious and aggressive pathogens, usually have thick mucoid capsule in their structure. Another example, some pathogens defend themselves against phagocytosis by producing what we call surface proteins. And those proteins just simply interfere with the contact between cell and phagocyte. As an example, we can use Streptococcus pyogenes. It causes strep throat, and this bacterium produces what we call M protein, surface M protein, and surface M protein interferes with phagocyte. It doesn't allow phagocytes to attach the surface of that bacterium. So phagocytosis is a part of the second line of our immune system. And microorganisms that survive phagocytes are then confronted by our third line of defense. The third line of defense, if you remember from anatomy class, produces antibodies against pathogens. So how pathogens avoid this third line, how they go around this third line of defense? So immune system recognizes molecules on the surface of microorganisms as antigens, and after recognition in a few days our immune system produces antibodies that are very specific for each specific antigen. To avoid this line of defense, some pathogens are able to perform what we call antigenic variation. What does it mean? It means pathogens change their surface antigens regularly, and then as a result we cannot recover from that infection. As an example, we can use Neisseria gonorrhea. It causes gonorrhea in humans, and gonorrhea is what we call not self-limiting infection. What does it mean? Self-limiting infection means infection doesn't require any specific treatment. We will recover from that infection without specific treatment. As an example, we can use common cold. We get infected, develop symptoms, in a week or so we recovered. But gonorrhea, for example, is not self-limiting infection. Why? Because Neisseria gonorrhea can perform antigenic variation, means it changes surface antigens regularly. As soon as our immune system produces antibodies against specific surface antigen on the surface of Neisseria gonorrhea, that microorganism, Neisseria gonorrhea, changes it again, and then antibodies that produced previously are not able to destroy new antigens, and our immune system has to produce new ones. As soon as new antibodies are produced, Neisseria gonorrhea mutates again and changes surface antigens again, and it happens constantly, and that explains why gonorrhea cannot be called self-limiting infection, because to recover from gonorrhea we have to go through specific antibacterial treatment. What else pathogens can do? Some pathogens can secrete what we call immunoglobulin A proteases. Those are enzymes that destroy immunoglobulin A, and those are antibodies that protect us from the surfaces of the mucous membranes. And the last example, some pathogens are able to block complement system. We will talk about complement system in one of our future lectures. It's a very large part of our immune system. It is a part of second and third lines of defense. Some pathogens have able to block, to actually prevent that system from working. All right, virulent factor number 6. Pathogens have to be able to multiply and reproduce within body of the host. And very often when pathogens multiply within the host body, they produce toxins. They can produce exotoxins, endotoxins, and toxic proteins. In this lecture, we're going to focus on exotoxins and endotoxins. Let's look at this chart. It compares the main characteristics of exotoxins and endotoxins. The first characteristic is chemical composition. Please remember, exotoxins are made of proteins. Endotoxins are made of lipids. Actually, it is a lipid A portion of LPS. And if you remember, LPS, it is a structure or substrate that is outer membrane of gram-negative bacteria is made of. So again, endotoxin, it is a lipid A portion of LPS. Produced by exotoxin can be produced by gram-positive bacteria and gram-negative bacteria. How about endotoxin? Since endotoxin is a part of the outer membrane of gram-negative bacteria, it means endotoxin will be only released when gram-negative bacteria get destroyed. Next characteristic, toxicity. Please remember, exotoxins are very strong toxins. They have high toxicity. On the other hand, the endotoxin is not as strong as exotoxin. It has low toxicity, but you have to remember, if your patient has bacterial infection that caused by gram-negative bacteria, you have to be very careful with the doses of antibacterials that you're going to use for treatment. Because if you use high doses of antibiotics, you will kill a lot of bacteria in a short period of time. And then a lot of endotoxin will be released into the bloodstream. And even though it has a low toxicity, but if a lot of endotoxin released, it can cause actually toxic shock in the patient. Immune response, next characteristic. Because exotoxins are made of proteins, they will be recognized by our immune system as antigens, and it means exotoxins can stimulate our immune system to produce antibodies. How about endotoxins? Endotoxins are made of lipids. Please remember, lipids are not recognized by our immune system as antigens. So that means endotoxins cannot stimulate our immune system to produce antibodies. And the last characteristic, diseases. A lot of infections, a lot of pathogens are able to secrete to make exotoxins, and as an example we can use tetanus, botulism, diphtheria. As an example of infection when endotoxin might be released will be meningitis. On this slide, you see a few examples of exotoxins that can be produced by different pathogens. For example, cholera enterotoxin. It stimulates intestinal epithelial cells to secrete a lot of fluids. That explains why those patients will develop severe diarrhea. E. coli enterotoxin. This enterotoxin is very similar to cholera enterotoxin, but not as aggressive. Tetanus neurotoxin. We call it neurotoxin because it affects nervous system of infected individual, and actually it causes rigid paralysis. Botulinum neurotoxin again. It also affects nervous system and causes flaccid or limb paralysis. The next one is diphtheria cytotoxin. We call it cytotoxin because it kills epithelial cells of the throat. And the last one is pertussis toxin. Pertussis toxin can irritate cough center in the human's brain. On this slide, once again, you see the main characteristics of endotoxins. So this type of toxins are called endotoxins because they are part of the cell's outer membrane. It's a lipid A portion of LPS. Endotoxins are released only when cell is destroyed. For example, when we use antibacterial drugs, antibiotics for treatment. And what they can do, they activate a complement system, they stimulate inflammatory mediators, and as I told you, even though endotoxins are not as strong as exotoxins, but if a lot of endotoxins are released in the bloodstream, they actually can can cause a toxic shock, collapse, and death. Alright, let's get back to exotoxins. Remember how exotoxins are made of proteins, and because they are made of proteins, they can be recognized by our immune system as antigens. When our immune system recognizes antigens, it makes antibodies. So we can say that exotoxins can stimulate our immune system. We use this knowledge to make toxoids. Toxoid is a type of vaccine which is made out of exotoxin. So basically we take exotoxins, we process them in the labs, make them not virulent, so they are not able to cause any symptoms of infection, but still are able to stimulate immune system. When we introduce toxoids, those vaccines, to the patient's system, it will stimulate the immune system of the patient to produce antibodies. Means patient will develop immunity against that specific exotoxin. So a vaccine against diphtheria would be an example of toxoid. On this slide, you see different types of viral infections based on pathogenesis. So based on pathogenesis, you can divide all viral infections into four main groups. The first group, this is what we call lytic infections. Let's look at this first picture of this slide. So this is our virus, this is our human cell, it's got infected, and then as soon as this infected cell starts producing viruses, it becomes provirus, right? It starts producing viruses. When they're mature enough, they leave that cell, and when they leave that cell, they destroy it. As you remember, naked viruses do these to their host cells, and that's why we call them mean or angry viruses, because every time when they get released from the host cell, they destroy host cell. Second type of viral infection is what we call persistent viral infection. Once again, this is our virus. Human cell is got infected, becomes provirus, and starts producing viruses. But when viruses are mature to leave their host cell, they leave it. And also, as they leave, they grab a piece of the plasma membrane and make envelope for themselves. Remember, envelope viruses do that. But host cell will not be destroyed for a while. It will continue making viruses. So that is an example of persistent viral infection. Next type, a latent viral infection. Once again, virus, human cell got infected. And remember, when patient gets infected with latent infection, at first it develops symptoms of infection and then recovers. But then that virus doesn't go anywhere. It stays somewhere in the body, within the body of the host, and every time when it gets reactivated, that patient will develop symptoms of infection again and again. Of course, as an example, we can use herpes simplex virus. And the last group of viral infections, those are oncogenic infections. You remember that some viruses can cause mutations in human cells, and as a result, those patients develop cancer. As an example, we're going to use human papillomavirus, HPV virus. It can cause cervical cancer in women. So that is an example of oncogenic viral infection. And finally, the last virulent factor we're going to cover is we're going to talk about how pathogens leave body of the host and return into reservoir. That means we're going to talk about portals of exit. For a lot of infections, portals of exit are the same as the portals of entry. For example, respiratory tract infections, sexually transmitted infections, infections that are transmitted a parenteral mode of transmission. But for gastrointestinal infections, it's always different because for gastrointestinal infection, usual port of entrance will be mouth, and portal of exit for gastrointestinal infections will be anus. We completed the second part of our lecture, and the last part of our lecture, we will talk about epidemiology. So epidemiology is a study of occurrence of disease. It means epidemiology studies when infections happen, where they happen, and how they are transmitted. From epidemiology point of view, all infections can be divided into 4 main groups. The first group is called epidemic infections or epidemics. What does it mean, epidemics or epidemic infections? It means we have outbreak of infectious disease that affects a lot of people within a short period of time. As an example, we can use flu or cholera or poliomyelitis. The next group, those are pandemic infections or pandemics. Pandemics, those are worldwide epidemics. As an example, we can use AIDS, HIV infection, or swine flu, for example, would be an example of pandemic infection. And then endemic infection. Endemic infections always present in population. And the level of those infections is pretty much the same all the time. For example, gonorrhea. We have outbreaks of gonorrhea here and there in different areas of the United States, and we always have a few people infected here or there. And the last, those are sporadic infections. Sporadic infections only occur occasionally. For example, during the war, a lot of soldiers will be wounded, and those wounds will be most of the times contaminated with the soil. But remember, soil is a source of Clostridium species, including Clostridium tetani. So that means large group of injured soldiers will develop symptoms of tetanus. Alright, one more term you have to remember from this lecture is nosocomial infections. Nosocomial infections, those are hospital-acquired infections. What group of patients usually will be affected by nosocomial infections? Of course, number one group is going to be immunocompromised patients. Then the second group, patients that go through invasive medical procedures, for example, surgeries. And then the last group will be patients that go through the treatment with the broad spectrum antibiotics. As an example of nosocomial infection, we can use, of course, urinary tract infection. Actually, it is number one nosocomial infection today in a hospital for catheterized patients. Another example will be surgical wound infections, pneumonia, or skin infections.